As the leading company for non-university research, Upper Austrian Research is a key player in the research, technology and innovation policy of the province of Upper Austria. In cooperation with influential partners and 10 highly respected associated companies, it supports the implementation of innovative solutions at the crossroads where fundamental and application-oriented research meet. At the Center for Advanced Bioanalysis, CBL for short, the latest methods in biophysics are made available to the next generation of medical diagnostics. Methods of biophysics and cell biology are developed for individual evidence in food technology, the pharmaceutical industry and medical diagnostics, where they can be used in immunology and cancer research, for instance. In this, the procedures used include microstructuring, surface structuring and functionalization, ultrasensitive microscopy with resolutions in the nano range, cellular model systems, customized assays to obtain individual evidence and specially developed biochips for the manipulation of minute amounts of fluid for the analysis of single molecules. Complex disorders require more complex diagnostics. We can address this problem with the methods we develop at the Center for Advanced Bioanalysis. The challenge is to make them useful and available for medical practice. With our core competences of miniaturization and highly sensitive analysis, we can make these tests smaller and more cost-effective. This means that in future, examinations tailor-made to individual patients will be affordable. Research findings in the field of biology have shown that the heterogeneity of cell populations play an important role in their function. This presents a challenge in cancer treatment, for example, since resistant subpopulations can survive chemotherapy, which can lead to a relapse after apparently successful treatment. Microfluidic biochips developed at the CBL make it possible to analyze the composition of heterogeneous biological and medical samples at single-cell resolution. The knowledge gained from this about the coaction of the individual cells can subsequently be used to develop improved approaches in the treatment of cancers, for example. The so-called microfluidic biochips are produced in the clean room. The chips are nanolabs the size of a postage stamp. With them, single cells can be analyzed. The chips consist of a small glass plate and 2 silicon layers, inside which microstructures for channels, reaction chambers, valves and pumps are produced using photolithography techniques. The sensor surface of the chip is created on the glass plate in the form of a microarray by a spotter. To address these complex issues, they have a multidisciplinary team, for whom service plays an important role, and a wide range of equipment at their disposal, including a combined atomic force and fluorescence microscope for the examination of biological cells. The biology laboratories provide customers with a wide range of possibilities for experiments and analysis in the fields of cell and molecular biology. For instance, customers nourish cell samples in the cell culture and dye them for analysis with fluorescent DNA dyes or antibodies. The size and particle size of each individual cell, as well as several of their fluorescent characteristics, are then measured using flow cytometry. This enables measurement of physical, biochemical, or functional properties of subpopulations in heterogeneous cell populations. Additionally, the defined subpopulations can be sorted for further analysis according to their characteristics. Every human being is unique, and the same applies to diseases. This means that personalized diagnostics and therapy will become increasingly important for effective treatment in future. This is where we see our chance and our task.